Hey guys, welcome back to the channel. So in this video we are going to discuss more about the subject environmental biotechnology. So let's get started. So previously I've made two videos for nitrogen cycles, so you may check them out. So starting with today's video, so moving on with this. So as we know, the nitrogen cycle mainly consists of many of the enzymes and lots of bacteria and microbes that help in the successful uh, endless going of the cycle. All right, so there are lots of bacterium, including free living ones, which can be of aerobic, anaerobic ones. Under aerobic we have nostoc and azobacter, and under anaerobic we have chromatium and chlorobium. So we'll talk about it in detail, and also we'll look on to some of the reactions or some of the steps that are involved in the nitrogen cycle in a broader sense, and actually understand what are its importance and how does it happen. So with this, so talking about the process of ammonification. So previously we talked about what is nitrogen fixation. All right, so we have talked about all other microbes and the enzymes that are involved in the first process or the first initiation of the cycle of nitrogen cycle, which is nitrogen fixation. So nitrogen fixation is nothing but conversion of atmospheric nitrogen to ammonia. All right, which is known as nitrogen fixation. So we have talked about the previous video of nitrogen fixation. So we'll just start off with the second process, which is ammonification, and how does we deal with it. So talking about ammonification, so decomposition of organic nitrogen from excreta, tissues of dead plants and animals into ammonia is called ammonification. Alright, so basically the decomposition of organic nitrogen from excreta and tissues of dead plants and this into ammonia. So basically this is retrieving of ammonia, or retrieving back the ammonia that was previously converted. So basically all the detritus or all the dead decaying matter that are present in the atmosphere are slowly gets converted back to ammonia, which is known as ammonification. The organic form of nitrogen needs to be broken down to ammonia, and ammonia later converted that can be reused by the plants. So basically the ammonia that has been retrieved in this process called ammonification gets back into the cycle of nitrogen cycle, and thereby the process continues and the cycle goes on and on. So it definitely involves some of the microbes that help in the initiation of this process, which includes various fungi, prokaryotes, which can be actinomycetes, bacillus like B. vulgaris, B. mesenterillus, and B. ramosus. Also, these secrete extracellular enzymes, which can be protease, lysozymes, nucleases, to degrade organic nitrogenous molecule in tissues. So yeah, definitely these two and just one more step, to correlate this. So definitely, as we know, we have just studied about these bacteria and these enzymes. So in every cycle, whether it be nitrogen cycle, whether it be water cycle, whether it be carbon cycle, and any other cycle, definitely just not the microbes do the work. Along with microbes, we need the cooperation of the enzymes, which help in the successful completion of any process. So in this we have some of the microbes here, and along with that we have some of the enzymes that help in this conversion process to degrade back into ammonia. So basically these nitrogen containing dead decaying matter gets converted to ammonia with the help of these enzymes along with these microbes. Also, this process liberates a lot of energy, which can be used by saprophytic microbes, so saprotrophic microbes. All right, so this is the overall reaction, as you can see. So all of these things release nitrogen along with lots of energy, which is 736 kilocalories, which can be used by saprophytes, and thus urea can be also, from urea we can extract ammonia. So this is another process as well. So moving on with this, we have another slide, as you can see on the screen. So ammonification involves basic two steps. So it actually involves two steps, which are proteins from dead decaying and waste products get decomposed to form amino acids. All right, so these dead decaying matter gets first converted to amino acids through decomposition, and these amino acids are later ammonified or gets degraded back to ammonia. Alright, so these amino acids return back to ammonia. And this ammonia, being gas, can rapidly disappear from the dry soil, but in moist soil it becomes solubilized and forms ammonium ions. Definitely in moist ions it combines with water or hydronium ions to form NH4 plus. Alright, so moving on with this. So talk about the second steps process, so the first step that we studied in the previous video, which was nitrogen fixation, then we talked about modification, and then we have nitrification. So talking about nitrification, which is carried out by chemoautotrophs. So what are chemoautotrophs? So chemoautotrophs are unlike any other autotrophs, but the difference is they depend on chemical substances for their, or basically they make chemical substances for feeding others, for feeding the producers, consumers mainly. Yeah. So these are carried out by chemical autotrophs. So this nitrification process is nothing but conversion of ammonia or ammonium into nitrate. So this is a two-step process also. So this is also a two-step process. So it all starts with ammonium or ammonia. All right. So starting with ammonium, which is definitely a toxic compound. All right, which slowly gets oxidized to form nitrite first. So the first step is formation of nitrite, NO2 minus. All right, which also liberates a lot of energy, and also it is present in soils and water of moderate pH. And some of the enzymes and some of the microbes that help in this process are which are of species such as Nitrosomonas, Nitrococcus, Nitrosospira, and accumulation of nitrate inhibits Nitrosomonas. All right, so this is one of the points that has been given. Accumulation of nitrogen causes inhibition of Nitrosomonas. So we have talked about some of the species as well now. So moving to the second step of it, which is conversion of nitrite, which is formed in the first step, which gets then converted into nitrate, which leads to liberation of small amount of energy. Right. So nitrite is toxic, as you can see, whereas nitrate is safe. All right. And the second step is carried out by the species known as Nitrobacter. All right, and Nitrobacter depends on Nitrosomonas to generate nitrite. So here comes the importance of Nitrosomonas and the concentration of it in the soil. So the amount of Nitrosomonas is very important, or the species of Nitrosomonas is very important for conversion of nitrite to nitrate. All right, so accumulation of nitrite won't be a advisable process because it will inhibit the working of Nitrosomonas. So Nitrobacter depends on Nitrosomonas to generate nitrite, and most plants and microorganisms can take up either nitrate or ammonium, and it requires oxygen and neutral soil pH. So moving on with this, so talking about the term which is nitrogenase. So what it is. So we'll talk about nitrogenase. We have also talked about this term nitrogenase in the previous two videos, so you may check them out. Also, nitrogenase is a very important enzyme that has some of the important components that are present. All right, so let's get started with this. Giving you a brief about nitrogenase, which is basically these are produced by certain bacteria which are mainly cyanobacteriums, and these enzymes are responsible for reduction of nitrogen to ammonia. All right, and this is very much applicable to the process of ammonification, which involves the same process of conversion of nitrogen to ammonia. Also, these nitrogenases are the only family of enzymes known to catalyze the reaction. All right, which is the key step in the process of nitrogen fixation. So which is one of the important steps. So nitrogenase is one of the important enzyme also involved in the process of ammonification. All right, so talking about some of its complexity, as you can see on your screens, which is it is composed of two major protein components. All right, the component one is 1-MOFE protein or molybdenum-ferrium protein, which has a molecular weight of this much, and second is 1,2-Fe protein, which has a molecular weight of this much. So this has two components, as you can see, which has two major protein components. So talking about some of the steps that are involved with this nitrogenase. Step 1 is ferredoxin reduces, reduces Fe protein, and second step is the Fe protein reduced binds to ATP now, and Fe protein reduces molybdenum ferrium protein. ATP hydrolyzed and MOFE protein donates electron to atomic nitrogen. So this is how the entire process gets done. So this nitrogen gets converted to ammonia. So these are the 4 steps that you may like to remember or take a screenshot of it, and which might come in your exams. So basically it just starts with a reduction of Fe protein. Ferredoxin reduces Fe protein, and this reduced Fe protein binds to ATP. So this reduced Fe protein, which has just been reduced, helps in the binding to ATP. Alright, and the third step is the Fe protein reduces MOFE protein as well, or molybdenum ferrium protein as well. So this does the job again. So basically it first binds to ATP and then it reduces MOFE protein. Alright, and then the Fe protein, which is there which was binded to ATP, gets hydrolyzed, and MOFE protein donates electrons to atomic nitrogen. So the last step is the ATP, which is hydrolyzed, and MOFE protein donates electrons, so which is now reduced, donates electrons to give rise to nitrogen. Donates electron to atomic nitrogen so that the conversion can take place. Yeah. So these are cyanobacterium, usually carry the nitrogenase enzymes in specialized structures called heterosites. So this is one important thing which might come in your MCQ. So these are stored in specialized structures known as heterosites or cysts. Alright, so these nitrogenase enzymes are stored in cyanobacterium in a specialized structure known as heterocysts that provide anaerobic conditions for fixation. Alright, on with this. So talking about the last step that we have here is the denitrification step. Alright, so talking about the denitrification step, as you can see, overall overview of this is this. So basically nitrate gets reduced to nitrogen, or atmospheric nitrogen is gained back with the release of lots of energy, as you can see, from nitrate. So under anaerobic condition, alright, so this is applicable for anaerobic conditions only and does not require oxygen. Nitrate is converted to gaseous nitrogen compounds, which can be nitric oxide, nitrous oxide, or inert nitrogen gas by microorganisms. Alright, so this is prevalent only in anaerobic conditions which do not require oxygen, in which the nitrate gets converted to nitrogen with the help of microbes, and it balances atmospheric nitrogen fixation. And denitrifying bacteria include species in the genera of Micrococcus denitrificans, Thiobacillus denitrificans, Pseudomonas aeruginosa, and Paracoccus and alkali genes. Alright, so this nitrate first gets reduced to nitrite, and this nitrite can be reduced to nitric oxide or nitrous oxide, which then thereby gets reduced to nitrogen with the release of lots of energy. Alright, so this was the process of nitrogen cycle. Alright, so we just starting. Previously we had studied about, the previous video studied about nitrogen fixation, and this we have started with a modification, then we studied about nitrification, then we are studying about denitrification. Alright, so moving on with, so these are some of the examples for this. And this we have bogs and swamps contains a lot of denitrifying bacteria, which basically convert back to nitrogen. These contain a lot of nitrites, as you can see, these soils, and hence their soil is deficient in nitrogen or nitrates. Plants there have evolved over time to consume insects to fulfill their nitrogen requirement. Alright, so basically they consume insects or different sort of species to fulfill their nitrogen requirement. Also, this is another process for your knowledge, which is Anamox process, which is anoxic ammonia oxidation. So anamox process, all right. So this is a coupled anaerobic oxidation of ammonia, all right. So this is a coupled anaerobic oxidation of ammonia and reduction of nitrate. So as you can see, basic thing is the ammonia gets converted back to nitrogen, all right. So this is can be said as a modification as well, or we can say the reverse process of nitrogen to ammonia is nitrogen fixation, as we know. So basically what happens is coupled anaerobic oxidation of ammonia takes place in reduction of nitrate. It enables NO2 removed from sewage plant effluents. So it's a double process, as you can see, in which ammonia gets converted back to or gets reduced to nitrogen, which we know as a modification, or nitrogen can be oxidized or fixed with the nitrogen can be fixed, which is very known as nitrogen fixation or nitrogen reduction. All right. So this is the two-step process that occurs simultaneously in the absence of oxygen. All right. So this is the anamox process in the absence of air, which enables the conversion to and fro of from ammonia to nitrogen and nitrogen to ammonia. So let's just keep this video till here. I'll be back with another video very soon. So stay tuned and thank you for watching this video.